Okay, so Spogazi, Khutlelelo, Zanele, we are working with you on this question. All right. The diagram,
hello.
we're going to start. Yes,
Hello.
I'm listening.
Okay. I am at work, so I'm just listening to my earphones.
Okay, let me see who's that. Zanele. Okay, noted. Noted. Okay, Spogazi and Khutlelelo, that means it's just the two of you then. Yay. Can I start?
Yes.
Okay. The diagram below represents a DNA molecule undergoing an important biological process. Okay, this is the molecule. We are able to see something here. So please do help me out. Identify the process shown in the diagram. What process is shown in the diagram?
Simple guys, I'm not sure, ma'am. I think
Okay.
it's DNA. Okay, DNA.
DNA what exactly?
I'm not sure because I didn't write one point.
Okay, we're here for you guys. If you didn't write it, that's fine. We just need to try our best in our activities. So if you didn't, that is fine. Then let's just listen into the lesson so that we get our answers. Is that fine?
It's fine.
Okay, so I did hear someone saying DNA replication. That is correct. I'm not sure what's happening with my pen, but I'm trying to write DNA replication. Thank you very much. Okay.
Please note me. You called my name.
Yes. Thank you, Kotlelelo. Thank you. But you are helping with the whole question, not just one question. Yes.
Okay.
I think I
weak hydrogen bond.
I
don't know.
Even
okay,
me.
let's take turns between. Okay, let's do this. Let's take turns between Spogazi and Khutlelelo. So one question for Spogazi, one question, the next question for Khutlelelo. Is that fine?
It's fine, ma'am.
Yes.
Okay. All right. So now Spogazi, this one is for you.
It's a weak hydrogen bond, ma'am.
Thank you so much. Weak hydrogen bond. My pen is failing me. So if you are able to hear me, please listen to what I'm hearing. Wow. Please listen to what I'm saying. If you can't really see what I'm writing, I'm not sure. I've just changed my pen. I'm hoping this one is going to work. All right. Khutlelelo, give the collective name for the parts labeled 2, 3, and 4. Where are they? So there is 2, there is 3, and there is 4. What is the collective name there?
I think it's nucleotides.
Thank you. Nucleotide. Okay. If the rest of you guys that are not answering, you want to ask a question, ma'am, why are we saying it's nucleotide or whatever, please feel free to unmute anytime and do ask a question. This is not limited to the two people that are answering. I think that's just for order. That's it. Then give the full name of the nitrogenous base labeled 5. There is nitrogenous base labeled 5. So what will be the full name there?
Thymine, Sepokazi. Thymine.
Thank you, Thymine. And they said full name, hey? So please note, if you wrote a T here, that would be wrong.
Yeah.
If you wrote a T, yes, because now the question said full name. So we follow rules in life sciences. If you don't, we penalize you. So they said full name, so the full name will be Thymine. If you wrote T, wrong, you lose a mark. Okay. 1.4.3, during which phase of the cell cycle does this process take place? Yes, team, we listening?
Okay. I'm not sure, but I said interphase.
Interphase is correct.
Okay.
Interphase is correct. Khutlelelo, where in the cell does this process occur?
In the nucleus.
Thank you. Okay, everyone, a round of applause for Sipogazi and Kutlelo, please. Thank you, guys. Okay, next, Bonginkosi and Slindo. These next few questions are for you. So Bonginkosi and Slindo, you're gonna take turns in the next question to answer. Let's please work together. I like how fast we are moving. If you want to stop me at any second, please do stop me, guys, so that I can explain whatever you need me to explain. A proper protein synthesis question. The diagram below represents a certain stage of protein synthesis. They're telling you a certain stage of protein synthesis. And we remind ourselves that there are two stages. There is transcription and translation. Let's look at this. The sequence of bases is read from left to right. That is important. Now, those A, C, G, whatever, we are reading them from left to right. They tell you. Identify molecule Y. There is molecule Y. There it is. Bonginkosi?
It's mRNA.
It's what? I didn't hear.
It's mRNA, ma'am.
Thank you, mRNA. All right. Then describe the process that resulted in the formation of molecule Y. How many marks, guys? 6 marks. We are not running away from this question and saying, oh, it's a lot of marks. I don't remember. We are writing it and we are getting the full marks. We are getting that 6 marks. Now, I think it was Slindo, right? Yes, Slindo, we are listening.
I did not attempt that one.
Slindo, you're hurting me. I am big on answering these questions, and I'll tell you, in the marking center, as soon as learners see a question that has about 3 to 6 marks, I'm telling you, there's empty spaces most of the time, but there shouldn't be, guys. For everyone in this tutorial, we're not going to leave empty spaces. We're going to attempt every question. Okay, I'm just going to ask you a question. Then I'll get someone else to give me the 6 marks. I hope that is fun. Now, our question here, just give me a second. Our question here is saying, describe the process. This is a copy and paste from the notes actually, and yes, it's for 6 marks. They're saying we must describe the process. They don't give us the name of the process, but they're saying it's a process that resulted in the formation of molecule Y. And we said molecule Y is mRNA. So maybe, just for me, can you give me the name of that process? They're asking us to describe for 6 marks. They didn't ask us to name it, but I want you to name it for me. Please. It's between these two. These two stages of
transcription, ma'am.
Thank you so much. You see, that's where it starts. So they're asking you to describe, guys, transcription, but they didn't put it that obvious in a question. Oh, my pen. They're asking you to describe transcription. But what do you then do? You have notes on transcription and how the process takes place. You then go to those notes and then you copy and paste those points. That's it. Now here they didn't make it obvious for you. They said the process that resulted in the formation of molecule Y. We know the process that resulted in that mRNA being formed is transcription. That's what they are asking you to describe. Now, the points in transcription, remember I mentioned them and I said there is a way that you need to answer and the order is very important. I'm not sure if I'll be able to write all of them, but we have them on the notes, guys. Point number 1, you speak about the DNA double helix unwinding. So in order for you to get that 1 mark, the first mark, DNA double helix unwinds. Please listen to me, guys. I'm not sure what's happening with these pens. Unwinds, that's the first tick. So it unwinds first, and then point number 2, you speak about the hydrogen bonds. The hydrogen bonds between the strands will break, the molecule unzips. You already have 3 marks by saying that. So if you then speak about the hydrogen bonds, you write it in full, and you say they break, and then you say the molecule unzips, you already have 3 marks, believe it or not. And after that, remember our transcription, we are going to use one strand. You say one strand is going to be used as a template. One strand is going to be used as a template. In order to form, this is point number 4, in order to form a messenger RNA strand. Now it will use free RNA nucleotides. Remember, when we speak about the nucleotides, you need to identify the nucleic acid. Free RNA nucleotides in the nucleoplasm, whereby A will pair with U and C will pair with G. Even if you leave it at that, believe it or not, you already have your 6 marks. And this is in our notes. We are not avoiding any question at all, especially if something is in our notes. Yes, I'm listening.
I wrote double helix unzip and weak hydrogen bond breaks to form 2 template strand.
Okay, read the first one for me, please.
The first one,
Yes.
double-stranded DNA unwinds.
Wrong.
And point number 2,
It's wrong. We need the word DNA. Last week, I made dots to say you have to mention these words in order for you to get a mark. So if you're saying DNA unzips, you're not getting a mark at all. Even if you say DNA unwinds, or if you say double-stranded molecule unwinds, wrong. We need the words to go together. That's how strict they are. DNA double helix unwinds. Those 4 words together give you 1 mark.
Okay.
Is that okay? Yeah, I'm listening. You have another point?
No, I'm fine.
Okay. So I want us to practice those points. Unfortunately, they will ask you to describe the process, but you don't know which process they're going to ask. Is it DNA replication? Is it transcription? Is it translation? Let's copy and paste those words into our mind. If you rephrase them, or if you rephrase them or you omit certain words, you're going to lose a mark. I'm gonna try and see if I can project them quickly. Okay. There you go. I'm trying now. It should come in less than 10 seconds on your screen. Okay. Okay, just give me a second. There, I can see them on my side, but I don't think they are clear yet on your side. So just give me a second. DNA replication. Oh, okay, I skipped it. Oh, there you go. Okay, I have just shown you guys the transcription slide. Please confirm if you can see it on your side. Thumbs up if you can see the transcription slide with the points. Thank you so much. Now, this is what I emphasized last week. And it's very important. You don't follow it, we lose marks. But this 6 marks, guys, please, 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 we are getting it. Now, the section of DNA, you don't necessarily have to write that, but this is what is important. DNA double helix unwinds. There's a tick. Then the word weak doesn't have to be there. Hydrogen bonds break between the DNA molecule, or hydrogen bonds break, you get a mark there. And the molecule will unzip. Sorry, the molecule unzips after the bonds break. So we can't start by saying the DNA unzips. That is wrong. So that's how technical they are in marking. It will need to unwind first, then the bonds break, then the molecule unzip to form two separate strands. That's the order. And then I said one strand will act as a template to form messenger RNA. Now, this DNA template will be used as a complementary strand. This strand of DNA, we are going to use it in order for us to form bases for the messenger RNA strand. Now, another tick will be here. This is done by using free RNA nucleotides. Please tell me you remember to write RNA nucleotides. And where do we find them? We actually find them from the nucleoplasm. Then we say the messenger RNA strand is complementary to DNA. Even if you end it here, you get your 6 marks. We mark the points separately, but they need to be in a specific order. If I ask you now to tell me how to bake, well, that's a bad example because I don't know how to bake, but you're going to say you mix this and that, and then you put in the oven, and then you check. You are telling me the steps of baking. It's the same thing with DNA transcription. You need to give us the correct steps. You can't say, put the flour inside the oven, then take it out, mix it with water, and then add whatever. That's wrong. If your steps are incorrect, your cakes are not going to be proper. It's the same thing. That's how it works, guys. Okay, let's move. I hope it's clear. It should be clear. I hope it is. Okay, I'm happy to take questions any second. I'm back to my notes, so I can't see what's happening on your side. Okay, we are still working with Bonginkosi and Slindo. All right, let us continue. Give one structural difference between molecule X and molecule Y. Just one. But let's look at the marker location. They are asking for one structural difference between the two molecules, and the marker location is 2. So that means you need to tell us something about molecule X and then molecule Y in order for you to get the 2 marks. Bonginkosi, we are listening.
Okay. It's X, it's double helix molecule. Then Y is single strand molecule.
Correct. Single stranded. Double stranded. You get 2 marks. And in a test, please do write. Can you see what they've done here? They've said molecule X and molecule Y. This is your molecule X, which is a DNA. And molecule Y is a messenger RNA, meaning it's a type of an RNA. So essentially, you are comparing between DNA and RNA. In your answer, you need to indicate, as I've done, X is double-stranded. You are telling us that X's DNA is double-stranded. If you were to answer and say 1 is double-stranded and 1 is single-stranded, you're not going to get a mark. Tell us about what they've asked. They've said X and Y. So you include that in your answer. Okay, Slindo, I'm sure you can give us another difference. What's another one that we can take note of? We listening, Slindo? Okay, Bonginkosi? It's still a partnership. So Bonginkosi, please do continue.
I think the nitrogenous base is the molecule X has the thiamine
Yes.
containing thiamine, then the molecules Y contain uracil.
2 marks. full 2 marks. Molecule X has thymine, you write it in full, right? And then molecule Y has uracil. That is the difference. You are comparing the difference there. You can speak about pairing of bases. I'm adding here. So molecule X will have base pairs because remember there are 2 strands and then there will be a pairing of bases here. And molecule Y is a messenger RNA strand. It's single-stranded. There is no pair there. There are no base pairs, right? You can also speak about the weak hydrogen bonds, the bonds that we find between the 2 pairs or are the 2 strands of DNA. And there are no weak hydrogen bonds that we find in a messenger RNA strand. My thing that I want you to take away from this is the marker location and the identifying of the molecules when you are asked. If they say X, you write that in your answer. Not one has this, one has that, you lose both marks. Okay, let's see if we can get another partnership to keep us going. Dipolelo and Sindiswa. Let's continue with you guys. Let's give Bongikosi and Lindo a break. So Dipolelo and Sindiswa, please do take over. Let's look at our question. Give the sequence of bases at triplet Z on strand 2. Woo. I need to erase a little bit for this one. So my screen is gonna go and come back. Just give me a second. I'm erasing. Please. Okay. It's taking forever to erase, but anyways, give the sequence of bases. They want the sequence of bases from triplet Z on strand 2. There is strand 2. Let me just change my color. There is strand 2 and there is triplet Z. That's what they are asking us. And remember the question said we read the bases from left to right. Okay, Dipulelo, what did you say?
I wrote ATG.
ATG.
G.
Okay, let me write it here. Option A, ATG. Anyone with a different base, anyone, it doesn't have to be the pair, but let's hear from the pair. Was that Dipulelo was speaking?
Yes.
Okay, Sindiswa, what did you write? Sindiswa?
Hi, ma'am. I didn't write, but I can pay.
Please tell me.
So I'm pairing with like HGA. Ne?
Yeah. So they need bases that are here. The bases that are here. So give me your 3 bases here at Z.
It's TC.
TC.
TCT.
TCT. All right. Thank you, Sindiswa. Okay, anyone else with a different triplet? Please just unmute and tell me. Anyone else with a different triplet? I saw a few hands. Devo.
So I have TAC.
TAC. I'm happy to take the last one. Thank you, Devo. I'm happy to take another option. Please do unmute and tell me what you wrote.
ATG.
ATG. ATG. And I heard an AUT.
UG.
Okay, is there another one?
AUG, ma'am.
AUT.
AUG.
Okay,
AUG.
okay, I'm not sure if I'm hearing properly. AUG.
Yes.
Okay.
Yes, AUG.
Okay. Let me drink water for this one. Yo. Oh. Okay, this was, I think, November 2024, if I'm not mistaken, 2023 or 2024. Let's understand transcription. So I want you to lead us into getting a correct answer. We said this strand over here is the messenger RNA strand. And they've told us that between these two DNA strands, this strand of DNA and this strand of DNA, they've told us that strand number 1 is the template strand. Remember when I taught protein synthesis, I said there will be 2 strands, right? In transcription, but we are only going to use 1 strand. In DNA replication, we use both. So now, if this is a strand that we are using to form our messenger RNA, this is called a template strand. But now, this template strand was initially paired with the other strand that we don't use, which we call a non-template strand. So what they have done here, they have given you the strand that was used, which is strand 1. They've told you that's the template strand. In order for you to find Z, you first need to find bases of the template strand. So let's look at it like this. If this is our messenger RNA basis, UAC, I want you to first tell me the DNA bases that were here pairing with UAC. Anyone can unmute and tell me.
It's. Can I answer, ma'am?
Yes?
Okay. It's A,
Yes?
A, T,
Yes?
G. Yes.
G. Okay. That's not our answer. Thank you so much for answering. That is not our question answer. We haven't answered
Ma'am?
2.1.4. So what I was saying was, let me get to the answer, right? So the basis that we've given here,
Ma'am,
it's
I
not
wanted to
what we,
answer.
I didn't hear.
I said I wanted to answer your question. You said you want us to give you a answer for AU. Can you repeat your question so that I hear you properly?
Oh, okay. Maybe there's a delay in sound because there is a student that already answered. So I got the basis. Are you able to see there in orange? Here. Are you
Yes.
able to see them? Okay, yes. So I've been answered already. So I'm going to ask another question. So you can, it's fine. You can leave your mic unmuted. I'm going to ask another question. So remember they asked us for Z. So I'm going there. But we need to remember that in order for us to get Z, we need to get bases for the DNA template strand that we used to form these bases in our messenger RNA. Now, those bases are ATG. We need to remind ourselves that these bases here were initially paired with the bases that we have there at Z. So you are not going to look for bases that pair with UAC, but you look for bases that are pairing with what? ATG. Why is that? Because this is a template strand and this is a non-template strand from DNA. This is a DNA strand, this is a DNA strand. Now, what you're seeing in between is a messenger RNA strand that used bases from this template strand. So you are going to pair ATG. When you pair ATG with another DNA strand, that will be TAC. Now, which one is correct from all of these? This one is the only correct one. Are we fine? Are we fine, guys? Everyone is quiet.
Ma'am, can I ask you something?
Yes,
You say the correct one is TAC.
correct.
So I wanted to understand, since they've been asking for Z, which is UAC. How did you get C? I mean, I understand A is pairing with U, and then A is pairing with T, and then how did you get C? Because you're saying answer is TAC.
Okay. I'm gonna re-explain it, but since I'm struggling to what you call this, erase, I'm gonna just remove this document and then bring it back. So just give me a second. Just want a clean document. It should be back on your screen. Let me go to Teams and see. Yeah, it should be back on your screen. Thumbs up if you can see it. It's the same question, but it should be clean now. Okay, thank you. Okay, I want us to go together, guys. Let's move step by step. Let's recap. When I taught you transcription, I said we will have 2 DNA. Wow. Something has stopped my pen from working. Okay. Technology, but I'm sorting it out. Okay, 5 seconds, it should be fine. Yes, when I taught DNA, I said you'll have 2 strands. Maybe let me write my strands in a horizontal form instead of vertical. So these 2 strands and these 2 that I'm going to write here are the same thing, just the pen is acting up. So this one strand is a DNA strand, and this one strand is a DNA strand. Now, between these 2 DNA strands, we are going to use one. And here we are using this one. The one strand that we use, we call it a template strand. We are going to use this strand in order for us to form a messenger RNA strand. Now I want us to work together, guys. So I'm going to forge my bases here. 1, 2, 3, 4, 5, 6. I'm forging them and I'm going to label them as T, C, A, G, A, T, right? Remember, this is a DNA molecule. Now I want us to work together, guys. The bases on the second strand of DNA, if this is a T, this would be an A. Are we together? Yes, yes?
Yes, ma'am.
Yes.
Thank you. Let's
Yes, yes,
unmute,
ma'am.
guys, so that I can hear you. So I want us to, I'm going to say the bases, but I want you to be there. When I'm saying are we together, yes, I want us to get this. So then we have a C. A C would be paired with a G. A will be paired with the T. G will be paired with the C. A will be paired with the T. Shout if I do make a mistake. It's 8 o'clock after all. So T will be paired with A. Are we fine so far?
Yes. Yes, ma'am.
Thank you. Then we said from these 2 strands of DNA, one is a template, and I'm going to say that's the one we are going to use to form a messenger RNA strand. The other is a non-template strand. We are just saying we are not going to use it. We are going to only use the template strand. Then from the template strand, we are going to use free RNA nucleotides that will complement the bases that are exposed in the template strand, there is this messenger RNA that we are going to form. And we are forming it using the exposed bases of the template strand. So that means T that is exposed will be complemented by an A. And I'm going to label it and say this is our messenger RNA strand. T will be complemented by A, C will be complemented by G, A will be complemented by U, G will be complemented by C, A will be complemented by U, then T will be complemented by A. Are we fine so far?
Yes,
Yes,
ma'am.
ma'am.
Thank you so much.
Yes, ma'am.
Okay, then let's move. What are we having here? Let's look at these bases over here. And these bases over here. This is what they've asked you. So what did they do? They are asking you for the bases that are in the non-template strand. They are these bases. And what are you given? You are given the messenger RNA codons. That's what you are given. You are given UAC. They are asking you for Z. Where is Z? Z is in the non-template strand. So what did I do with the answer? I said, you cannot pair these ones. You cannot pair a messenger RNA codon with a non-template strand. So where do we need to start? In order for us to get the basis of the non-template strand, we need to start here because this was the partner. So this T and C and A, that's what we need to get in order for us to get the A, the G, the T, if we are not given. So in our example, they wanted Z, and Z is bases in the non-template strand. How do we know that this one is non-template? They've told us that this other one is a template strand. So the same example, they gave us UAC. So we cannot pair this UAC with a non-template strand because that's not how it works. So we need to find the bases that complemented UAC, and those bases was A, T, and G. Now that we have A, T, and G, we're like, okay, where is your partner? This is what we found, we need this. So this is now what we have, we need this, which was paired initially with the template strand. Now these are the bases of the non-template strand. We look, A was obviously paired with what? With T. And T was obviously paired with A, and G was obviously paired with what? C. That's how we find TAC. Are we fine?
Yes, ma'am.
Okay.
Yes,
Protein
ma'am, but
synthesis.
it's confusing, hey?
I know.
But
I
we get
know.
it.
And
I
they
get
did it
it.
on purpose. I am telling you, I marked this paper. It was either 2023 or 2024. I can tell you from the batches that I marked, there was only 60% of kids that got it. Everyone else was getting it wrong because what most learners did, you looked for bases by just looking at the messenger RNA strand. So you went, okay, I have UAC and they are looking for the bases at Z. So we are going to say, okay, U will be paired with A will be paired with T since the DNA, and C will be paired with G. Most learners said there's ATG, which was incorrect. But it will take a little bit of practice. Okay, let's do 215. I don't even remember who was helping us now.
Okay, ma'am. Can I
Yes,
ask
I'm
you,
listening.
I just wanted to know, how do you know is a non-coding? How do you know it's a non?
They told us. Let's look.
Oh, they told us.
Yes, let's look here. When they say this is a template strand, that is the coding strand. That's the one that we are going to use to form messenger RNA. They told us here. They said strand 1 is a template strand.
So a template strand is a non-coding.
Template and coding is the same thing. So template strand is a coding strand. The reason we're calling it coding, we're going to use that code. The code is the sequence of bases, that TAGC, whatever. That is the code that we're going to use to form our messenger RNA. Hence, it's called coding. Then, strand 2, we call it non-template. If you don't want to use the word non-template in a test, they can say non-coding strand.
Okay.
Happy?
Yes, ma'am. Thank you.
Oopsie. Thank you so much. Let's answer this one. I want to hear your answers for this one. 215. The table below shows some amino acids that are coded for by codons. Remember, when we say codons, we are speaking about bases in which molecule? Tell me. We call 3 bases a codon. In which molecule? Anyone? Three bases in which molecule? Atli? In which molecule? Is it in a DNA, in a messenger RNA, or in a transfer RNA? 3 bases that are called codons. Okay, anyone, guys? I'm not moving up until you give me an answer. We're not moving.
mRNA.
Messenger RNA.
Thank you, messenger RNA. So the table below shows some amino acids coded for by codons on molecule Y. Remember, you don't need to cram this table. Whatever table you get, you will be given in a test. So you don't need to cram that, oh, okay, this code or these bases code for this amino acid. You don't stress about it. Identify the first and the last amino acids coded for by this section of molecule 1. This is very important. And remember, how are we reading the bases? From left to right. And they are saying they want us to identify the first and the last amino acid. One amino acid is determined by 3 bases. So what are we having here? We are having codon number 1, codon number 2, and codon number 3. We need a name for codon number 1 and the last one, since we read from left to right. Now, the codon that we have as the first one is AGA. So it's AGA. We are going to look for this AGA in the table. There it is. AGA codes for which amino acid? Arginine. You write it down in full. Arginine. You mess up the spelling, wrong, because it's a copy and paste. Arginine. So we are done with the first one. Then the last one, this is the one. CCA. We don't need to complicate anything. We are going to take these 3 bases, CCA, and look for them in the table. Let's see. Where is it? There is CCA. What is the name of the amino acid? Proline. I'm happy to take questions. Raise a hand if you've got these 2. Please do raise your hand if you got them. I'm not seeing hands.
Got it right.
Okay, I do see a few hands. Okay, okay. I'm happy, guys. For those that did not get the amino acids, please do tell me that you are now getting it. Please do tell me if you are now getting it. Is it a thumbs up if you are fine and you're getting it? Okay, all right. Thank you, guys. I think this is good that we do these activities now so that once it's exam season, we should be really in a good space. I'm going to move through this one just to save time, but if there is a question, just unmute any second and ask me. Now, you are given 2 diagrams there, which are showing 2 types of nucleic acids. I'm hearing someone, I'm listening.
Yes, I just want to ask a question.
Yes.
So here on the 2.1.5, when we answer this question, is that necessary to write AGA before AGA arginine, or we can just write the full name only?
Wow, I can't tell you how important that question is. You write the name only. If you start by writing A, G, A, and then arginine, we can penalize you. Thank you for that. You write, you just give us the names of the amino acids. This is what you write. Then we mark you. Now, if you just write AGA and CCA and then the name, if they apply the rule of the first answer only, they are going to mark AGA and they say that's wrong. That's not the name of the amino acid. Then they mark CCA and say that's wrong. That's not the name of the amino acid. So that's why when you started asking, I was like, okay, thank God you asked. Let us write the answers. If you want to figure out your answer, figure it out on the question paper and then just give us an answer. Do not start by writing the basis and then your answer. It's not maths. Thank you so much for that. I hope it's clear for everyone. Woo! Yeah, that was important. Okay,
Okay.
let's go through this one. Is it sorted now? Mulalo?
Yes, yes, yes. I wanted to know that.
Okay, thank you. That was a very necessary question. I forgot to mention it. Okay, now you are having two diagrams. You are given diagram 1 and diagram 2. I'm going to just go through all of this. Identify the nucleic acid in diagram 1. Diagram 1 is definitely a DNA molecule. So we've answered this one. That is a DNA molecule. And then identify A and B. There is A. A is our hydrogen bonds. Remember, you don't need to write weak hydrogen bonds. The word weak is not necessary. Hydrogen bonds is necessary. Then B, there is our B here. That is actually the phosphate group. Remember, in a nucleotide, you get a phosphate group, a sugar, and a base. So that is a phosphate group. Even if you write phosphate, you get a mark. You don't need to write the word group. Name the type of sugar that is found in the nucleic acid represented in diagram 2. Remember, we are able to see that, okay, diagram 1 is DNA and diagram 2 is RNA. So the type of sugar that is found in RNA is a ribose sugar. That is a ribose sugar. The tick will be here at ribose. Whether you write the word sugar or not, it doesn't matter. Which diagram? 1 or 2 represents a nucleic acid that is used for tracing of human ancestry. Now we use DNA to trace that. So that means it is diagram 1. Since we said diagram 1 is DNA. Then the next question says, which diagram 1, which is DNA, 2 RNA, represents a nucleic acid that is used for paternity testing? That's obviously when we do tests to confirm if the male is the father or not. We actually use DNA. So that is diagram 1 also. How many marks here? 6 out of 6. I'm happy to take questions. I'm listening if you have a question. Just 1 minute for questions for this. This was straightforward. I think if you did get confused, it would probably be just there at the end with the tracing of human ancestry if you got confused. But I think everything else was clear. I'm happy to take questions. 3, 2, 1. No questions. We move. That was easy.
Oh, question. Our question again.
Yes, we are
I have
listening.
question also.
Yes, I'm listening.
Here, it says which diagram 1 or 2 represent a nucleic acid that used for tracing of human ancestry. So if I just write only 1 without mentioning diagram 1, is that correct?
That is correct.
Okay. Okay,
That is
thank
correct. Okay, like the questions. Okay, then let's move to the next one. Ooh, it's another protein synthesis. So that means I'll be asking questions. Okay, the diagram below represents a stage during the synthesis of a certain protein. Who? They are saying it represents a stage. Remember, we have two stages, transcription, which is the first one, and translation, which is the last one, or the second one. That means the diagram that we are given represents either one of these stages. What are we able to see here? We are able to see amino acids. These are amino acids. We are able to see this molecule over here, that shape of a molecule is a type of an RNA. We're able to see a transfer RNA. We are able to see this sequence here, or this strand that is attached to a certain organelle. Now, it's very easy for us to know what this is, and it's very easy to know what this strand is from. Okay, what it is and where it is from. Okay, let's try and work together. Identify organelle 3. This organelle here, that is the ribosome. Ribosome. Please, like I said, do listen to what I'm saying because my pens are not doing the greatest of jobs today. So that one mark, I don't want to lie, you can't mess it up. That one mark, all of us should get it because that is a ribosome. I spoke about it last week and I said the messenger RNA strand, remember this process takes place in the cell. Then it's the nucleus whereby the first stage is transcription. Then the messenger RNA will move out of the nucleus and attach itself to the ribosome. So this is what we are talking about in this question. Describe for 2 marks. We're not running away from this. Describe the role of molecule 1 during this stage of protein synthesis. Now they are asking you to describe the role of this molecule. But before you can be able to describe it, you need to know what it is. And this molecule is transfer RNA. That's the only molecule that has that kind of a shape. Now, it's very easy for you to describe this, guys, but remember what I said. Whenever you are asked to describe, you need to think in your mind, what molecule is that? What am I describing? And then you give the answer. If you start by telling us that that is a transfer RNA, you're not going to get a mark. Now, the main function of a transfer RNA is to bring amino acids in the cytoplasm. Why are we saying it's to bring amino acids? Remember, I'm saying this is a transfer RNA. What is it carrying here? At the top, it's carrying an amino acid. And here at the bottom, it carries anticodons. These 3 bases here, these 3 bases anticodon, that's the transfer RNA. So it's actually busy. In front, or rather here at the bottom, it carries the anticodons. Then in the back, it's the amino acid. And now remember, the anticodons are the ones that are determining the amino acid that is being carried. For 2 marks, how do you need to answer this? Is to bring the required amino acid to the cytoplasm according to the messenger RNA codons. To bring, remember I said my pen might not write perfectly, so listen to my answer. To bring, what will it bring? Amino acids. Where is it bringing these amino acids? To the cytoplasm, or you can say to the ribosome. To bring these amino acids to the cytoplasm or ribosome. That is the 1 mark. But now these amino acids that are brought, it's not just any other amino acid. It's influenced by the codon of the messenger RNA. Now, remember, this is the messenger RNA. I'm giving away an answer here. So now the amino acid that is brought here is determined by these codons over here. These codons will need to complement this anticodon that comes with that amino acid. That's how it works. So the 2nd mark will be according to the messenger RNA codons. That's the 2nd mark. 1st mark, is to bring the amino acid to the cytoplasm or the ribosome where this stage is taking place. And the amino acids are brought according to the messenger RNA codons. Okay. Give the sequence of nitrogenous bases at 2.
Oh.
I'm
Ma'am?
listening.
Can I ask you something? I wanted to know if I'm right when I said the tRNA transfer amino acids and arranges itself on the mRNA.
Read it for me again.
Okay, tRNA transfer amino acids and arranges itself on the mRNA.
You might get 1 mark. Transfer amino acid. Can get 1 mark for that. But bring to the mRNA. They are not bringing to the mRNA. The amino acids that are brought to the ribosome are determined by the messenger RNA. So that last sentence,
Arranges
though. A.
I said and arranges itself on the mRNA.
Okay, wait. What arranges itself now?
The amino acids.
Okay, let's read it again.
I said tRNA transfer amino acids and arranges itself on the mRNA.
The second part is incorrect because we are saying it arranges itself. The transfer RNA doesn't arrange itself. It's all about bringing the amino acids, and those amino acids are going to be bonded together. It just brings the amino acid for it to be bonded with the rest, and the amino acid that is brought is determined by the codon of the messenger RNA. The minute you say it arranges itself, it doesn't line up and arrange itself. So they read the English into it. So unfortunately, because the wording is there, but then the sentence itself becomes incorrect. So then you lose a mark. I might give you 1 mark for that only.
Okay, thank you, ma'am.
Yes, Rita? Rita, are you sorted?
Yes, ma'am, I'm sorted. Thank you.
Thank you. I'm sorry, guys. The marking is very strict. The marking is very strict. I heard someone else. Yes?
Ma'am?
I'm listening.
Yes, ma'am. I just want to ask if the way I wrote it is correct or not. So
Okay.
on my side, I wrote bring the required specific amino acid. That's the first one. Is that correct?
That's 1 mark.
Okay, and then the second one I said according to mRNA codon to the ribosome.
Second mark. That's it.
So both of them are correct.
Yes. Both of
Okay,
them are
thank
correct.
you.
Okay, I'm gonna be one to say that life science is very unfair in terms of marking. We know it, we discuss it all the time as teachers, because you need to read into that statement that you're given from our notes and understand it. The minute you try to rephrase it, it means something else. So let's try that. It's gonna be a bit hard for us to follow what has been said on the notes, but let's try that and make sure that we understand properly what we are saying. If I'm saying I'm going to school, and I'm going from school to home, that means 2 things. Because of that word from and to. I am going to school, I'm going home from school. It means 2 different things. That's how they always argue it. So it's all about making sense in your answer. So, well, I'm going to be harsh in terms of telling you that your answer is incorrect. That's the only way you're going to learn to write the correct answers and phrase it nicely. Because in the exam, that's the same rule actually. I could be marking your paper in May, so well, I wouldn't know if it's your paper or not. Okay. I think at 2, are we done at 2? No, we didn't answer that. Give the sequence of nitrogenous bases at 2. There is 2. 2 is the anticodons here that are going to code with GGA in the messenger RNA. So that means your anticodons here, G will be complemented by C, G will be complemented by C. A will be complemented by U. Please tell me you got this one. Can I get thumbs up if you got this one? This one is easy. I'm seeing 2 thumbs up. I'm seeing 2. Okay, 3. Okay, 4. Okay. Okay. Guys, I need everyone to be getting this. This one is not bad. Okay, a few thumbs up. Okay, I'm happy with that. All right. Then let's see. Okay, give the sequence of nitrogenous bases on the DNA molecule that coded for histidine. Oh, what is this now? This is an amino acid. And where are we going to find this amino acid? We are going to find it here. There is the name of the amino acid. What exactly do they want? They want nitrogenous base on the DNA molecule. On a DNA molecule. Already they want just those 3 letters, but how many marks are they allocating to? That means you need to work a little bit. It's not going to be a copy and paste like this one. You see the one where they said it 2. We were just pairing with what we see and they've allocated 1 mark. So there's a theory and a lot of things that we consider in terms of allocation of marks. Now, I'm interested to hear what you guys got. Okay, let me take 4 options. I'm excited. Okay, A, anyone, give me your bases. Anyone. I'm going to take 4.
So I got CA.
Okay,
C-
CAT.
CAT.
And then I got GTA.
G.
Yeah, GTA.
G. T. A. Okay, anyone else?
I got CCA.
C. C. A. Last one, any other different base? Okay, so we have 3 options. I want us to remind ourselves of something. The sequence of bases, we look at it from a DNA molecule, and then we get a messenger RNA molecule, and then we get a transfer RNA molecule, and then we get amino acids, and then we get a protein. That's what we've discussed. Let's see what we are given here. We are given the amino acid and the anticodon. This is the amino acid. We can write it down. We don't necessarily have to. Histidine. That is the name of the amino acid. And what are we given? The anticodon, which is GUA. What is the question asking? It's asking for the DNA triplet. It's asking you for the bases that we would find here. I'm sure now you can work it out. So if in transfer RNA you are having GUA and you are asked about DNA, you need to step back once and get mRNA first, which are the bases you will get here. But once you get these bases here, you then need to use that to get the basis for what? For DNA. So the basis for messenger RNA that would complement the anticodons would be C, I hope I'm getting it right, CAU. Okay, now we have CAU, we are here. This is going to make it easy for us to then get the triplets in a DNA molecule. C will be paired with G, A will be paired with T, we are speaking about a DNA, and U will be paired with A. That is your answer, GTA. There it is. Tell me how you feel. Unmute and tell me how you feel. I'm listening, guys. Whether it's a sigh or yes or no or oh no, I need something. Divu, your hand is up.
Got it right.
You got it
Yes,
right.
yes,
Yay.
ma'am. Yes, ma'am. But this one was tough for us. It was tough for us because we spent like around more than 30 minutes trying to find out what the answer is. But we end up having the correct answer. But we fight a lot. It wasn't easy to get it.
Now you have it. Because let's see what's happening here. I can guess that people said, okay, there is the amino acid, there are the bases, they're definitely asking for these ones. So then you were complementing these ones. You went G, will go with C, U will be paired with A, and then A will be paired, since you are assuming it's DNA, you will say CAT, hence this option. Another person could have said, okay, this can be a U. So other people probably said CAU. That is what they were not asking this. They said in the question, DNA molecule. This molecule is a messenger RNA. So you have to take it back like this for 2 months. There will be no obvious protein synthesis question. They will try and trick you and trick you and trick you. But you need to show them that you're good. You've got your content knowledge, and it doesn't matter what different styles of tricking they're going to try on you, you'll be fine. So I hope it's clear now. I can see that the mood is going down a little bit because people are just probably thinking, I'm getting it wrong, I'm getting it wrong. You will get it right. It takes a little bit of practice. You will get it right. Okay. Oh, we did not speak about mutation, but let's see if we can answer this one. So the last one, if you didn't get this one, it's fine because we're going to address mutation. I hope I will have time. During a synthesis of the same protein, during the process of making the same protein that is made above, there was a change in the amino acid sequence because alanine was replaced with tryptophan. Now, this is speaking about mutation. Now, mutation is the change that could possibly happen in the bases. And this is what I want you to think of. Normally, when we speak about mutation, other people will say it's a change, other people say it's a mistake. Now, it's a mistake or a change that occurs in the sequence of bases. It normally happens during DNA replication or meiosis. Now, whatever change that occurs in the nucleotides or the sequence of bases or the genetic code will have an impact on what happens in the messenger RNA codons and will have an impact in what happens in the transfer RNA anticodons and will have an impact on what happens in the amino acid, and ultimately it will have an impact on what happens in protein. Now that's the theory of it. I haven't started answering. That's the theory of it. So I want us to look at it like this. If we were to have a DNA base TAC, obviously the messenger RNA codons would be AUG, and the transfer RNA anticodons would be UAC. That will code for a certain amino acid. I am just forging an example here, which is a protein. Now, a sequence of these different amino acids that is an amino acid, which will code for a protein. Now, if there is a certain change whereby this base here, C, is replaced by a G. That's the mutation that we are speaking about. Now, the change that is going to occur here, I want us to trace it. Now, if we are saying C is replaced by a G, let's look at what happens. We initially had TAC. If C is no longer there, we then end up having TAG. Now, since there was mutation in the DNA bases, that will have an impact on the rest of the other components. It will affect messenger RNA, transfer RNA, and probably the name of the amino acid and the protein. Let's look at our messenger RNA now. Since our new triplet is TAC, that means The new messenger RNA codon will now be AUC. Check, it has changed. It will also change the anticodon because now the anticodon is UAG, sorry. UAG, and UAG can code for a different amino acid, maybe a glycine. So what has happened here? A change in DNA impacted messenger RNA, which will also impact the transfer RNA, which will also impact the name of the amino acid formed and ultimately the protein. Because of what? C was replaced with a G. That changed everything. Let's go back to our question. Now, that's a bit of mutation. I haven't covered everything, but that is a bit of it. Now, what are we having here? They're saying during the making of the same protein, there was a change in the amino acid sequence. So where did the change happen? They're telling you about a change that occurred here. Now, in the change of the amino acid, they are saying an alanine was replaced by a tripotine. So instead of my example whereby I said C was replaced by a G, they are saying an amino acid alanine was replaced by amino acid tryptophan. Explain the possible reason for this. Why would an amino acid be replaced? Now you are now taking it back. I started from DNA and I was going downwards. You're going to start from amino acid and go upwards. Or you can even move from amino acid and say, actually, there was a change in a DNA base. That 3 marks we are not ignoring. Now, explain the possible reason for this. You need to start by saying mutation occurred. That is the change. Mutation occurred. Now, when this mutation occurred, what happened to everything? Now, the mutation occurred, which changed the basis of DNA, which resulted in a change in the codons. Check the terminology. I speak about bases when I speak about DNA. Then I say it changes the codons of messenger RNA, which then changed the anticodons of transfer RNA, which resulted in alanine being replaced by tryptophan. I'm becoming specific. I'm not saying amino acid being replaced. I'm referencing what has been given here. Hectic. You can even work out the bases here. But I don't want you to stress about the bases. How would you get the bases now? You are given the amino acid. There is amino acid alanine. There is amino acid tryptophan. You need to get the DNA bases for this CCC. Remember, this is messenger RNA. You need to get bases for this, and then you get bases for that. So you'll then tell us that, okay, what were the bases here? How have they been replaced here? So how I have it here, GG, a change in the sequence of nitrogenous bases from GGA to GGG to GGA, sorry. Where is this GGG from? GGG. Where am I getting this GGG? These are bases in a DNA. What did I use? I used messenger RNA. Then for the DNA bases of these codons, that is G and AA. So these are the DNA bases, and this is the DNA base. So if we replaced tryptophan with this, so that means our initial base was GGG, which got replaced by GAA, that is the mutation. Then you continue with the sequence. This is another one that needs a little bit of practice, but you should be able to get it after some time. I'm happy to take questions on this. I know this can be a bit too much, but I'm happy to take questions. The mood is very low today. Group A, you guys are always vibey. Okay, Ndivu?
Okay, so could ma'am please repeat? So what I have is mutation occurred, which changes the base of DNA. So what comes after that?
Okay, let's look at this sequence. So if there's a change in DNA, That will cause a change in what? We here? Are we here in Divo?
Oh yes. In the
Okay, so you need
sequence.
to lead me because I've written it down. So if there is a change in DNA, that will cause a change in what?
In the sequence
Of what?
of the messenger RNA.
Messenger RNA what?
I'm not sure what to say now.
Messenger RNA codons, terminology. So if there is a change in the DNA base, it will change the messenger RNA codons, and then that will result in a change in transfer RNA anticodons. So what happened here? They've told you the names of the amino acid. They said alanine was replaced by tryptophan. Let's go up. There is alanine and there is tryptophan. What do you have for alanine? You've got your anticodons, you have your codons. You need to get your DNA yourself. So this is the DNA triplets for the initial amino acid, GGG. How do I get GGG? I used the codons from messenger RNA. Same, second name of the amino acid, tryptophan. So we have our anticodons, we have our codons. I need the DNA. So you make your own DNA using these codons, GAA. So initially we had GGG, which then got replaced by GAA. That is why this amino acid is replaced by this amino acid. You'll get it. This one takes a little bit of time. I don't want to lie, but we'll get it. I'm going to skip this one, guys. Describe transcription. We've done it already for 6 marks. So we've done it already. Let's come here. Okay, it's another protein synthesis. The diagram below represents a process that occurs during protein synthesis. Identify molecule X and then molecule Z and then molecule, okay, just X and Z. Okay, so we should be able to identify what is happening here. It represents a process. If we are seeing a transfer RNA, that means this process is translation. There's an I there. So they've asked us to identify X, that is the amino acid. Then they've asked us to identify molecule Z. That is easy. There is Z over here. That will definitely be messenger RNA because that's obvious for us. In a transfer RNA here at the bottom, it will carry the anticodons, and the anticodons are going to be complementary to the messenger RNA codons. So Z is messenger RNA. Excuse me, I hope that is clear. Okay, then we move to the next question. Give the nitrogenous base sequence of the DNA base triplets to the middle codon on molecule Z. Check the marks. How many marks? 2. So that means you have to work it out. It's not a copy and paste. So you need to work it out. It's not a copy and paste. So what do they want? Let's break it down and get the terminology. They want the bases of what? Of the DNA base triplet that is complementary to the middle codon at molecule Z. They want us to focus here. And they want DNA for the middle codon. Codon are the 3 bases in a messenger RNA strand. Let us look for it. Remember I said we read the bases from left to right. So if we were to read the bases, 1, 2, 3, this would be the first codon. 1, 2, 3, this would be the second codon. 1, 2, 3, this would be the third codon. The question said we must focus on the middle codon, which is this one. Now we can't copy AUG and paste it because AUG is a codon in a messenger RNA strand. They want you to give us the DNA triplets. So you look at that order of mine that are always right. Where do we start? We start by having a DNA, then a messenger RNA, then a transfer RNA, and then an amino acid, and then a protein. So what are we given? We are given a codon in a messenger RNA. The middle one is AUG. What have they asked you for? They've asked you for the DNA triplets. So you're going to use that to complement. AUG will be complemented by DNA bases TAC. Unless I'm wrong. You can tell me if you feel like this is wrong, then we can work it out. Let me just confirm. I'm correct. It's actually 2 marks.
Yes.
That's how you work it out. I'm listening. There was someone. Okay. Did you guys
Yes.
get it though? But now at least you can see how they're trying to trick you. Please thumbs up if you got it. Thumbs up. Thumbs up. 1. I'm seeing 1 thumbs up. Thumbs up if you got it 2. Okay. Okay. Just 2, 3, 4. Okay. Okay. We're getting there. We'll be fine. Name. I'm rushing. Y. Give the nitrogenous base sequence of Y. This one is easy. And how many marks? 1 mark. So how are you going to get Y? There is Y. Why are these anticodons here, which are complementary to the bases of the messenger RNA? So C will be paired or will complement G. A will complement C, U, A. So now that means the answer is G, C, A. I'm hoping I'm correct. 1 mark. It can be. Because A will be paired with U. Yes. Yes, yes, yes. Are we fine, guys? I made that mistake with the C, but no. It shouldn't be a C
Yes,
there.
ma'am.
Okay. Thank you. 7 marks. Huh? How many of us ran away from this question? I was like, I'm not even going to attempt it. I'm not even going to attempt it. This 7 marks, I don't know it. It's difficult. We don't run away from marks here. When we see these marks, we go, yay, I want to get these marks. Let's see what this question is asking. Name and describe. You're doing 2 things. You are naming and then you are describing the process shown in the diagram during the formation of protein. You name what you are able to see there. When we see transfer RNA, amino acids, anticodons, complementing the codons, that is translation. So we name the process first, translation. And then after naming, we need to describe it. So that means naming will be 1 mark and describing will be 6 marks from that 7 marks. Terminology is important here. So you name, that's translation. Then remember that slide where I said it's a copy and paste. It's written exam guidelines in your notes. It says the same thing that I'm going to read here. Each transfer RNA carries a specific amino acid. When the anticodon on the transfer RNA matches the codon on the messenger RNA, then the transfer RNA brings the required amino acid to the ribosome. Amino acids become attached to each other by peptide bonds to form the required protein. This is a copy and paste from the notes. It speaks about the transfer RNA. It speaks about ribosome. It speaks about the codons of the messenger RNA. The transfer RNA will bring the required amino acid with the anticodon that is determined by the codon of the messenger RNA. All of that is brought to the ribosome to form that required amino acid. The amino acids, these are the amino acids that are brought to the ribosomes. they are going to be bonded together by peptide bonds. This is a copy and paste from the slides. 7 marks. The table below, oh, that's another mutation. The table below shows the codons that code for some amino acid. Use this table to answer these questions. A mutation, when you see the word mutation, you think change. Change happened. A mutation caused a DNA-based triplet to change from CTG to CTA. Describe the effect of this mutation on the protein form. I want us to move together. We are going to do this together. In the normal sequence of events for protein synthesis, we start by getting a DNA sequence, and then we get an mRNA codon, and then we get a transfer RNA anticodon, and then we get amino acids, and then a protein is formed based on the chain of amino acids. Now, please make a note that this is an application question. You are not going to generalize to say a change in DNA will cause a change in messenger RNA, which will cause a change in transfer RNA, and then a change in amino acid. Oh, that long sentence of yours will grant you 0 mark. Why am I saying you're going to apply the theory? I said it's an application question, so you apply theory. They have given you the basis. So what are you going to do? You are going to tell us a mutation has occurred in the DNA triplets. From what to what? You mention it. Where are we moving from? We are moving from CTG. And when this mutation occurred, we then ended up having a triplet of CTA. You write it in your answer. The DNA-based triplet changed from CTG, which is what you are given, to CTA. Now, how will that impact the messenger RNA codons? Remember, if now the base was initially CTG, that means the codons were G. I have to think twice here. It's 9 o'clock. It is G, then it is A, then it's C. So that means GAC changed to become what? You're coding it with this. So I hope you can follow up what is happening. You can follow suit. So from GAC codon, we are now going to have a GAU. Then the transfer RNA. Initially, we are going to have a transfer RNA of CUG. But now, because of this base and because of this codon now, we will then have CUA. You're still not done. You have to work for that 4 marks. And I said it's an application question. So if we're now having this transfer RNA, what is the change for amino acids? We are going to use the table. Our table is in messenger RNA codon. So we're going to focus here. We need to look for the name of the amino acid that was formed when we had this codon, GAC. We look for it. There is GAC over here. So what was the name of the amino acid that was formed? Aspartic acid. So the amino acid that was formed was aspartic acid. But now, because of the change, it's no longer going to be aspartic acid. It will be an amino acid that is coded for by GAU. Let us look for GAU. There is GAU. But now GAU is coding for the same amino acid. So it will be aspartic acid to aspartic acid. You don't need to write that. You actually write that it codes for the same amino acid. If it was different, you would write the name of the amino acid to say the change was from aspartic acid to maybe, for example, valine, if the base was coding for a different amino acid. But now our bases, GAC, there it is, GAU, there it is. They are both coding for the same amino acid. Now, how does that change the protein? The protein will remain the same. Why is the protein remaining the same? Because the amino acid remained the same. So there was no change in the amino acid. So there is no need for it to be for us to get a change in the protein. That's your 4 marks. Guys, can I hear your voices? The mood is way too low just because of protein synthesis. I need comments, guys. Please unmute. I can't be alone. I'm hearing that laugh, Kotlelelo, because wow, your mood, guys. Only because of
It's
protein
getting-
synthesis.
Getting worse.
It's mooring us. Gemoer.
It's getting worse, man. Yeah. Ha, ha, ha. Yay, I wish I had your brains.
Phelile ukuphapha kuphelile.
Ina phansi. You, I feel so nje ayibonga sis. Oh,
It's stupid.
it's okay. It's
No. I was allowing you guys to vent because wow, you know, last week after the lesson, I was like, I really love this group. I don't want to lie, I do love you guys. The energy, the questions, like I can feel your energy and the vibes, and we're laughing at the end of the lesson. So I was very happy. But today, hi Shem,
No.
the sugar levels are very down.
I'm out.
We're not gonna be out. This one we are going to do, and we are going to get it. What you then need to do after this lesson, sometime this week, you start over. You do these very same activities without looking at the memo. I will send the memo without looking at the memo, without looking at your previous answers. And you need to address this with attitude and aggression to say, I'm going to get it. There's no ways.
Yes.
There is no ways. There were over 75,000 kids in Gauteng that wrote life sciences, and more than 73,000 passed it. So why would you struggle? You're going to pass. It's all about pushing. It's all about doing the right thing, and you study and study and study. So we are going to be fine. I'm hoping to get a nice question to pick up the energy level, but mutation is a bit tricky, but you have to apply. If you didn't apply all of this, wrong. Okay, but this one, we can get good marks here. I mean, energy levels. Let's unmute and get our energy levels a bit high. Let's see. So they are saying we must tabulate 2 differences between DNA and RNA. They've told you nucleotides, not just difference between a DNA and an RNA. They are talking about that nucleotide, that monomer that has a sugar, a phosphate, and a base. That's what we need you to tell us. You draw a table. So there's rules you need to follow. Those rules that I said I'm going to address in a Saturday lesson. Hopefully I'll have energy to do that, guys. I really want to do it for you. Then, in a table, you are going to give us the column headings to tell us, okay, in this column, I'm going to speak about this. In this column, I'm going to speak about that. Now, you are comparing between DNA and RNA nucleotide. You are going to write here DNA nucleotide. You're going to tell us information about it. And then this side will be RNA nucleotide. Then you give us the heading of the table. The heading of the table, you start by writing a table showing. What is the table showing me and you? You copy this part as is. 2 differences between DNA and RNA nucleotides. That is your table heading. And then you are going to write the two differences. The first one, you can speak about the sugar because that's the main difference. DNA has a deoxyribose sugar, and RNA has a ribose sugar. I want to check something. Raise a hand if when you were comparing, you wrote deoxyribose on one side and you wrote ribose on the other side. Give me a thumbs up. If you just wrote deoxyribose for DNA and you wrote ribose for RNA, give me a thumbs up if you did that, so that I know how many of you did that. Thumbs up, I'm seeing 2, 3. So, can I give you the bad news? If you wrote deoxyribose for DNA and you wrote ribose for RNA, wrong. We don't take one-word answers when we are comparing. What is this deoxyribose? What is this ribose? It's a sugar. So you have to tell us that DNA has a deoxyribose sugar, RNA has a ribose sugar. If it's not a full sentence, wrong. So let's work on it. Okay, the next one, we can speak about the base. And then you underline or you rule off to say, I'm done comparing. You then can speak about the base. DNA has nitrogenous base, thymine. That's a full sentence. If you just write thymine uracil, wrong. We don't accept one-word answers. Also, this side, you will then write has nitrogenous, base uracil. This side, nitrogenous base thymine. Then you have your marks. How do we mark this for 5 marks? The first mark is known as the T mark. If you draw your table and it has all your table lines and the heading, you get 1 mark. Then the rest would be for the correct answers. Then that will be out of 5. We're not going to finish all the activities. All right. So we'll leave it at that.
Can I ask before you move?
Yes.
I just want to ask if here on this one, the last one that you give us, if I wrote nitrogen bases on DNA, adenine, guanine, cytosine, and thymine, is that correct, or I need to write it like this?
What did you write on the other side for RNA for that comparison?
Nitrogen bases on RNA are adenine, guanine, cytosine, and uracil.
I can say 1, you can be penalized. I can also say you can be given a mark. Depends how they analyze it. Now, when you differentiate, highlight what is different. Now, between the 3 or between the 2, these 3 bases are the same. So that is not a difference. What we are interested in is those last 2 bases. So that's what we need you to mention. So that's why I'm saying 1 can say. Yeah.
Okay, sorry. I was saying just because of on both of them, I end on DNA. I end with thymine, and on RNA I end with uracil.
Uracil.
So
yes.
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 I
So I
don't know.
get that.
I just want to find out if I can get a marker if I write it like this.
That's what I'm saying. It's possible for you to get a mark. It's possible for you not to get a mark, right? It depends on how we analyze it. It's possible for you to not get a mark because someone can argue and say, you are mentioning something that is the same. Yes, your answer about U and T is at the end. You've mentioned them, which is what is different, but you've started by mentioning things that are the same. Why are you including those in your table? Now, if that is the argument, you lose the mark. But then if the argument is, No, this learner is identifying the 4 bases that we have in DNA and the 4 bases that we have in RNA. And from those 4 bases, we are able to see that the last ones are different. Then if the argument is that point, you get a mark. So it depends. For me, I would say, just tell us about thymine and uracil to be safe.
Okay, thank you.
Okay.
From now on, I will remove this other one. I will write nitrogen bases are thymine, and on the other side, I will write nitrogen bases are uracil. Is that
Yes,
correct?
please.
If I read it like that.
That will be more than correct. Okay. All right. We have a few minutes left, and I thought I was going to end at 8:30 today because I've had a long day from 6 o'clock. But I want us to finish off DNA. I want us to look at this. It should come to your screen. Just give it a few seconds. Now, mutation is an important part. I've touched on it. I'm gonna try and see if I can go through it for 2 minutes here. But what I want us to look at is the DNA profiling, which is an important part of this topic. So it should take me 10 minutes to speak about it, then we're done. So what you see on your screen is just different examples of protein synthesis and mutation. I've covered mutation. So if it's blank, just give it time. It's going to load. I'm just scrolling down. That's why it's doing that. Just give it a bit of time. Okay. Okay, this is what I want to speak about. Thumbs up if you have DNA profiling on your site. If you have DNA profiling on the notes, thank you. Thank you. Now, this section is important. It doesn't get assessed with a lot of marks. So we're looking at a maximum 7 marks for just this section and the topic. But they are very strict with terminology. So with terminology, they are super, super strict. So what is this DNA profiling? And you need to use the term DNA profiling and not DNA fingerprinting. Don't. DNA fingerprinting is that one, you know, when you go to the bank and you open an account, then they ask for your fingerprints. That's the one. We're not talking about that. Now, DNA profiling includes literally getting into a cell and taking a sample of a DNA. Yes, we can get our DNA samples from different, I would say, I don't want to say parts of our bodies, but different collection methods, right? So a hair strand, we've seen this on TV, or saliva, or they can take the blood and get your DNA. That's the DNA profiling that we're speaking about. The definition: a DNA profile is a pattern of black bars, almost like a barcode. So when they collect your DNA, they then use technology to be able to analyze your black bars. Now, those black bars are the ones that indicate the DNA that you have. And your black bars should be identical to you and no one else. So if they were to collect my DNA, my black bars and the sequence and the arrangement, whatever, it will be unique to me. It will be different to everyone that is here on the group, with an exception of twins, and specifically identical twins. So identical twins have the same DNA. So that means their DNA profile will be the same. But fraternal twins, identical twins are the ones that are the same gender and they look the same, meaning they were from the same sperm cell and the same egg cell. That is why their DNA is the same. Fraternal twins, they look different. They can be different in gender. They can be different in how they look. So those twins don't have the same DNA because they are from 2 different sperm cells and 2 different egg cells. That is why I'm saying our DNA profiles will be very identical to ourselves and not anyone else. Now, the profile itself, profiling is the process. Profile is the pattern of bars. And these bars are produced on an X-ray film. When they extract the DNA, they get a sample from a crime scene or a sample from a guy that is suspected to be a dad or whatever. They run it through a test and then we get these bars. Now, these patterns of length or whatever, or these bars are in different patterns and length and thickness according to whatever biological test that they do. Now, the chemical structure on everyone's DNA is the same. So the structure of your DNA is the same, but the only difference is the order of your base pairs. So if my base pairs are A, C, G, T, someone else's base pairs might be A, T, T, G, A. So that's the only difference between my DNA and your DNA. It is the sequence of bases. Now, millions of base pairs will exist in each individual's genome. Your genome is your full set of your genetic material that makes you you. Now, we look at a DNA profile for us to identify individuals, for us to determine genetic relationships. So they can get a DNA of someone that unfortunately died in an accident scene. They should be able to identify their name, surname, and then get a hold of their family members. So that is what happens here. That's just the background. Okay, so how do we do this? I'm here. So how is this done? DNA is going to be analyzed for unique sequences to identify individuals and genetic relationships. I've mentioned that. How this is done, a DNA sample is needed to perform a test. So I've mentioned this. Our DNA is identical except the identical twins. Identical twins. If you just say twins, it's incorrect. A DNA sample is needed to perform this test. So you need a DNA sample in order for you to check who committed a crime, a sample from the potential father, a sample from the child, a sample from the mother in order for you to perform a test. So you can obtain a sample from blood, from hair follicle, from saliva. You can get DNA from semen, from the skin, or any cell. Remember, all cells will have the DNA. So you can get a sample from that. Now, in order for you to be able to perform this test, it will require 2 samples because you need to compare. You can't compare something that is just alone. So you have to compare. So now, what are some of the uses of this DNA profiling? So this can be used to identify paternity. Make a star here. They will ask you about the uses of DNA profiling. This is easy 2 marks that you can get. So they can use it for paternity testing to determine the father of the child. We can take blood tests for the mother, the child, and the father. They can use it in forensics to identify organisms from their remains. A body that is old, that is decomposed, damaged, or whatever, they can run tests on different cells there. They can identify crime suspects in forensic investigation. In a crime scene, they can get samples and then identify the suspects there. They can trace missing persons by using their DNA. They can identify genetic disorders. The only thing you need from this slide is the words that are in bold. That's it. If you write the word that is in bold, you're able to get a mark. To identify family relations, your siblings, your cousins, they can use DNA to trace all of that. So uses of DNA, it's easy 2 marks that you can get. You just mention the words that are in bold. Then there are limitations. Oh, this is also important. As much as we can use this DNA profiling method to be able to identify suspects, to identify paternity, to find family relationships, blah, 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 there are limitations to it. The reason there are limitations to it is because this is done by human beings. So human beings are the ones that are going to collect the samples. Human beings are the ones that are going to run the DNA into that biological test. They are the ones that are going to interpret all of this. So one of the limitations of DNA is that there is human error. This is 2 or 3 marks that we can get when they ask you about limitations or why these results can be considered incorrect. We can say this is incorrect because a human made a mistake. It's possible that this is not my DNA. I was not at the scene. That is possible. So human error is one of those answers because human beings may make mistakes. Short segments, if you collect, depends on the amount of piece of DNA that you collected. They believe if you collect a small amount, then your results might not be accurate. Now, uniqueness, expense, this process is expensive. There can also be contamination with other DNA. So for example, in that scene, maybe there was blood of two people that were mixed up, then they collect that. That means it will be incorrect. And it can literally be anything. I would say take human error, take short segments, take quality control, take sample size. Just choose 4 that you can remember. That's it. They're not going to ask you about everything. Then we look at this DNA profile. It's 9 o'clock, so I'm not sure if this is going to shut me out or what. Anything is possible. So with this, you read the bands or the bars. How are we going to do it? This is what a DNA profile looks like. This is a DNA profile. Now, how do we compare? We compare the position of 2 bands. So what do we have here? This is the evidence that was found in the crime scene. And this is the DNA of the 3 suspects. So we need the DNA of 1 or 2 or these 3 suspects to actually match the DNA that we find as the evidence. So you are going to compare the lines. It needs to 100% match. So how are we going to do this? Let's look at the first band. We call them bands or bars. Please make a note. Do not call them lines. Do not call them anything else. Bars or bands. So if this is the DNA that we find as the evidence, we need actually 1, it can't be 2 or 3 since it's identical. Only 1 person's or 1 suspect's DNA needs to match this. You are going to match the lines. So how do we match the line? Let's look at this one. Let me use a different color. I'm going to end the lesson in 5 minutes. I'm struggling to take it in now because I'm really, really tired. So let's look at this band over here. Does it match with any band in this position? You are looking at the same position. So I would advise you to use a ruler here. So in the same position, you can unmute. Do we have a band in suspect 1 in this position?
No.
No.
No. Do we have a band in this position in suspect 2?
Yes.
Yes.
Yes. So immediately suspect 1 is eliminated. Why is it eliminated? In order for suspect 1 to be the perpetrator, why am I saying his? Why can't it be a hair? Anyway, his DNA will need to 100% match the DNA that is our evidence. Meaning the lines of the suspect, every line needs to match in terms of the position. If it doesn't, then the suspect is not the perpetrator. So already we are eliminating 1, already we are eliminating 3. It might not be that obvious in a test, but let's look at the rest of the lines. What about the second line? The second line is matching suspect 1, but we've already eliminated because there was 1 band that was out. That means suspect 1 is incorrect. Anyways, we can even look at the rest of the bands. Then suspect 2, we are still winning. There is another band. What about the third band? Okay, third band in the evidence. There is no third band in the same position. Am I right? Yes. In the same position, there is no band here. And what about suspect 2? There is a band. Then what about the space here? There is a space and there's a space. So can you see that suspect 1 is eliminated because there is a band in this position for suspect 1. There is no band in the evidence. So 1 is out. I'm sure you can see what is happening. All of these bands here that you get in evidence A, in terms of their position and their size, is the same as all the bands that we get in suspect 2. So that means this suspect 2, guilty as charged. Easy peasy stuff. That is for forensics. Now, let's talk about this one. Paternity testing. We are identifying the father. Now, this is what I need you to think of. This is the only way you're going to manage to do this. Remember, the child will inherit 50% DNA from the father and 50% DNA from the mother. So whenever you are comparing paternity, you will be given the profile of the child, the profile of the mother, and the possible fathers. What does this tell us? That means 50% of these bands that we have in the child should match with the father, and 50% of those bands should match with the mother. But now, what is the rule and the easiest way to do this? If you look at a band in the child, you first look for that same band and position in the mother. If it's not there in the mother, it should be there for the father. It can't match for both. If you're able to see the same band for mother and father, it should belong to one of the parents. It can't be both. Now let's look at our child here. We focus on the child for paternity. So if we look at the child, there is the first band. Let's compare it to the mother's profile. Do we find the band in the mother's profile? No. So that means this section of DNA was inherited from the father. Which father? Are we finding anything with dead 1? No. Are we finding anything with dead 2? Yes. We can make a dot. What about dead 3? Yes. So dead 1 is eliminated so far. Why are we eliminating dead 1? Because this band here is not in the mother. If it was in the mother, we would say, okay, this band was inherited from the mother, but there's nothing in the mother. It should definitely be inherited from the father. Now we are only having 2 possibilities so far, dead 2 and dead 3. Then we look at this band, second band. Let's apply that rule I told you guys. A band is a section of DNA that is belonging to one of the parents, and you start with the mother. Then if this band, we find it in the mother, that means this section of DNA was inherited from the mother. We can't look any further and say, let's look for the same band from the father. It's not possible. It was inherited from the mother. We leave it. We go to the next band. What is happening here? This band over here is inherited from the mother because it's in the same position. There is the mother. We don't even look at what is happening to the father. Because what are we having here which can confuse you? You can say, oh, okay, there's a band in dead 1. There's a possibility. There is no possibility because this band, we can confirm that it was inherited from the mother. Let's look at this one then. This one here, what are you looking at? Okay, you are saying this one can be inherited from dead 1, but we've already eliminated dead 1. It's fine, we can look at dead 1, but there's nothing in the mother. Then let's look at dead 3. I'm not sure if it's still in the same line, but it does look like it's in the same line. There's dead 3. So we had this and we had that. Then what about this one? Where are we finding it? In dead 1, there's nothing. In dead 2, we're getting it. But then in dead 3, there it is also. So we're making a dot. Then what about the last one? We're finding it in the mother. Now let's look at the deads. Only one band matched in dead 1. Dead 1 is definitely not the dead. Yay. Dead 2, there were only 2 bands. Why are we crossing them out? Because dead 3 had how many bands matching? 1, 2, 3. You are the father. Help me, help me, help me, help me. There is the mother. This is the only activity we're going to do. There is the child. There's 3 males. Tell me, who is the dad here? Who is going to get a Happy Father's Day gift? Is it male 1, 2, 3? I'm giving you 1 minute. Time moves very fast. 30 seconds left. Remember, you focus on the offspring. You match the bands by using the offspring's bands, not lines, not fragments.
Can I ask the answer?
Yes, I'm listening. I'm listening.
My answer is dead too.
Definitely. Definitely. Thumbs up if you also think it's dead too, or if you guessed that it's dead too. Thumbs up. Guys. Or you still working it out? Yes?
Yeah.
Okay. It is male too. Let's look at it quickly, guys. Yo, I'm hungry. I need to eat. Let's focus on the offspring. There is a band. Please unmute. I think there's 8 of you now. Other people have left us. So please unmute. Let's finish proper, please. Okay? Then there is the offspring. Remember I said you say offspring and then you look at the mother first. Is there a band that matches this one from the mother?
No.
Yeah. No. So we are going to look for it in one of the fathers. Is it there in male 1? No, it's not there. So already we eliminate male 1. Is it there in male 2? There it is. In male 3, it's not there. We can eliminate or not, it's up to us. Then we look at this one. This band is there in the mother. We tick, we move. We can't look for it here in male 1 because it's already there in the mother. This one, it's not there in the mother, so we need to look for it in the father. There's a space in male 1 and in male 2, there it is. This band, it's not, okay, no, I'm skipping. Okay, no, I'm done with that one. Then this one, it's not in the mother, it's there in the father. This one, it's in the mother, we focus on that one, we focus on that one. Because both of them are in the mother, meaning this child inherited these bands from the mother. This one, it's in the mother, we focus on that one. This one is not in the mother, it's in the father there. 1, 2, 3. Boom, male 2. No, the energy can go up for this one, guys. Don't do me like that. The energy can go up for this one. This one is not